Hi everyone, Andrea Joss here. I'm talking to you today about protein, and this is the second class in the protein series. So today we're going to start to talk about all about protein, but in particular amino acids and peptides and protein structures and how they're made, etc. So we will get into those nitty-gritties here. Sorry. So first, before we do that, I just wanted to talk a little bit about the importance of proteins in our body. And the truth is that we can see and understand how proteins work in various different aspects by looking at how important they are in and around our body and for our body function. So first of all, 15% of our body weight is protein, and we can find them in many different places. One of the main places are structural. So a lot of our proteins are structural. They make up our bones and our muscles. And so those are part of the musculoskeletal system, which provides structure to our body. The most common protein there is collagen protein, but there's also several others. Enzymes are proteins. So some of our digestive enzymes, which you may have talked about already, like amylase, that is a protein enzyme. There are transport proteins that are either spanning the cell membrane that are within our circulation, like albumin or hemoglobin. And I'll show you a picture of hemoglobin structure a little later on in the lecture. They're protective proteins, so our immune system makes proteins and secretes proteins that act in protecting us. And so these are the antibodies. We also have proteins in our skin that are protective, as well as proteins that are in relation to blood clotting, like thrombin and various other blood clotting proteins that are quite important. Smooth proteins, so this is the non-contractile muscles. So some of our organs are made up of smooth protein. And then we have contractile proteins or striated, which is muscle. Protein-derived hormones that we can talk about, and I'll show you a picture of this one. Insulin is a very important protein-derived hormone. And there's also some of these large protein molecules within our plasma that act as fluid regulators. So they help to regulate fluid balance and osmosis, which is quite important when you're talking about electrolytes and other such factors. So proteins do help with fluid balance. And there are proteins that help with pH regulation, and these ones act as buffers. And we'll see here that there are various amino acids that are charged, which also help to improve our buffering capacity. It's really important for our body to stay within a particular pH as well as a particular temperature, but with respect to pH, amino acids help to balance the pH in our body. So I have this figure here as well, which kind of talks about proteins and types and functions that I got a couple of them that I mentioned already. And I just want to make the point here that proteins are really, really important for the body, as you can see here, but they're generally not used as fuel. So we use carbohydrates and fats a lot more as fuel for our body metabolism or during exercise, but generally speaking, our body does not like to use proteins as fuel because, as you can see here, there are so many other functions for proteins in the body, and protein is so important in many other ways. And this is not the case for carbohydrates or fats. So generally speaking, the body would rather use proteins for making other proteins, like muscle or any of these other types of proteins, rather than for fuel. Okay, so the basic building block of all proteins are amino acids. As I said here, amino acids are the building blocks of proteins. And these are some features of amino acids. So they all have this central carbon, okay? They all have an amino group on one side and a carboxylic acid group on the other side. So this is the backbone, if you will, of every single amino acid. What makes each of them different is the fact that they all have a different side chain or a different R group. It's sometimes called. And depending on the features that are within this side chain, this dictates the different properties of the amino acids. So these side chains could be polar, which means they interact with water and other ions. They could be nonpolar, which means they're hydrophobic and they don't like water. They could be positively charged or negatively charged. They could have rings on them. They could be very branched. In any case, these side chains are what dictates the function of the protein. And you'll see that this becomes really, really important when you're looking at secondary, tertiary, and quaternary structures of proteins and how they are formed, okay? Each protein is different, and each of them has its own 3D configuration, and this is brought about by these side chains and the interaction of the molecules within the side chains with other side chains of different amino acids that are in the same sequence, okay? So these side chains, these are side chains, as we'll see in the next couple of slides. They, again, dictate the different properties of the amino acids. And we can classify amino acids as either essential or non-essential, and I'll get to what this means in a moment. So here are these essential versus non-essential amino acids. So you can see here there's 9 essentials and 11 non-essentials, okay? So in total, there are 20 different amino acids, and this is how they're broken down. This is one of the classifications, essential and non-essential. And the next slide, I'll tell you what that means. We also have conditionally essential, okay? So that's these stars over here on the non-essential side. So what does this all mean, okay? So this is classifying these amino acids. So essential or indispensable amino acids, which they're sometimes called, is an amino acid that cannot be synthesized from scratch in the body and thus must be supplied by diet. So if we go back to the previous slide, you can see here that these particular amino acids are essential, meaning that our bodies cannot make them. So a lot of amino acids are made in the liver. In fact, these non-essential ones are made in the liver from time to time. But we need to get these essential amino acids from our diet in order to have the full complement of amino acids to make all the proteins that our bodies require. And you know, just to kind of plant this seed in your head so that, you know, you can think ahead a little bit or think back to the previous lecture where I discussed this, protein quality is really, really important. If we're consuming an average amount of protein, then it's really important that we consume a higher quality protein that has all of the essential amino acids in it, so that when we do that, our body has the building blocks for proteins. To make whatever protein it needs to. Okay, if you're eating a protein food that is deficient in one of these essential amino acids, this amino acid would be labeled as the limiting amino acid, and so this may actually limit your body's ability to create new proteins. Now, there's things that your body does to get that particular essential amino acid, like break down other body proteins and release it into the amino acid pool, which we'll talk about in a bit, but we don't necessarily want to do that because you want to maintain your muscle and maintain your protein as it's structured within the body, okay? So again, going over here, so the essentials are those that we cannot make in the body, we need from the diet. The non-essentials are the amino acids that can be synthesized by the body, and the conditionally essential ones are those that are usually not essential, which means that you can make them in the body, but there are specific times in which the body can't do that. So, you know, I've listed here in times of illness or in times of stress or potentially other times like, you know, maybe in pregnancy when metabolism is really high and you require a lot of protein, things of that nature. So conditionally essential amino acids, they are usually not essential except in times of illness and stress. And there are 6 of those that are conditionally essential, okay? So under special conditions, usually some pathophysiological conditions, okay? So most of the time it's in conditions of illness, as I've listed here, prematurity in the infant. So these amino acids are conditional in the infant. or individuals with severe catabolic stress, this is when the body cannot make these particular amino acids. Okay, so this is more of the case, not necessarily during normal circumstances, except for when maybe metabolism is much higher, which also occurs during illness, by the way. Okay, so going back to this figure then, these are the non-essentials. The stars are the conditionally essential ones, which under certain circumstances, they can become essential. Okay? Okay, so classifying amino acids. We classify these amino acids based on their side chains because remember, the backbone is exactly the same. Okay, we have the amino group, carbon group, and the carboxylic acid group is the same. The only thing that's different is the side chain. So different ways to classify these amino acids, and depending on how we classify them, this affects their function, as I said, and just as an example, it affects where these amino acids are within the body. So if you think of a lipoprotein, let's say, like a cell that carries, or it's a transport protein for fatty acids and cholesterol and other such things, which you will learn about in this class a bit later, because I don't think you've done the lipid section yet. In any case, if you think of a lipoprotein, you look at the structure of a lipoprotein, okay? You have proteins that span the lipid outside. You have proteins within the lipoprotein. And so depending on the side chain of the amino acids, these proteins are either on the cell surface because they have a polar side chain and so they can interact with water, or they have a non-polar side chain or a hydrophobic side chain, and so they're inside the lipoprotein. So this is just one example of where you can find proteins. You look at any membrane-bound protein, and you can see that the part that points outwards has the polar end, okay? So here are some ways to classify. So we have aliphatic and aromatic. So aliphatic amino acids are those that are straight or branched carbon chains. The aromatic ones have rings. Okay, so let's go to the next slide here. So we can see a couple of these, and you can see this particular figure. I put the source down here, but this figure separates them into some of these classifications. So if we go back, aliphatic and aromatic, there are only 3 aromatic amino acids. So let's take a look. We have phenylalanine. There's that ring over here, tryptophan as well, and tyrosine. So these are our 3 aromatic amino acids, and all of the other ones are aliphatic. So they're either, their side chains are either straight or branched. They don't have this particular benzene type ring. We can also classify amino acids based on their net charge. So amino acids are either neutral or are also called zwitterions. They are acidic or dicarboxylic and have a negative charge, or they're basic or dibasic and have a positive charge. So let's take a look at this for a moment. So here are the particular charged amino acids. Sorry, we have the negative charged ones down here, which are acidic, and the positive charged ones up here, which are basic. And so what we see here is the normal backbone of the amino acid has both a positive and negative end, okay? But then what makes these particular amino acids positive or negative charge is that there's an additional charge within the side chain, okay? So if we see here for the acid, the actual acidic amino acids, the ones that are negatively charged, we have aspartic acid and glutamic acid. Within their side chains, okay, they have additional charges, as we can see here on the oxygen. In contrast, okay, so these positive or basic amino acids, again, they have the positive and negative charge on the amino acid themselves, which all of these do, okay? But it's their side chain that carries the additional charge, which makes the total amino acid charge. So lysine, arginine, and histidine have positive charges, on an amine group within the side chain, and so this makes them positively charged. Okay, oops, let's go back here. You can also, again, classify amino acids based on polarity, and I've talked a little bit about this already. So whether they interact with water, electrolytes, minerals, etc. Now, these polar amino acids are not charged necessarily, so they don't have an acidic or basic charge, but they are polar, which means they can accept or donate hydrogen ions. So if we look over here, these are the non-polar versus the polar amino acids. We can see here that for most of the polar amino acids, they either have an OH group or a double bonded O or something of that nature where they're able to donate or accept hydrogen ions. Okay, so this makes them polar and it allows them to interact with water. These non-polar amino acids tend to have CH groups. Okay, they tend to have varying branched styles of CH groups within their side chain, which makes them not polar. Okay, so these are the hydrophobic amino acids, and they're usually found inside of a cell or inside of the lipoprotein, as I used as an example previously. Okay, so those are some of the different classifications. So take a look at this figure. I suggest you become familiar with the different amino acids and how they can be classified. You don't have to know what the different side chains are specifically, but you should know how to classify them based on these particular classifications. Just for your reference, here is another schematic that further classifies these amino acids. So again, classifying them into hydrophilic or hydrophobic. The hydrophilic ones are polar, which means they can interact with water. Hydrophobic ones are not. And within the polar group, we have the negative and positive charge, so the ones that are acids or bases. And then we have the uncharged polar molecules, or amino acids, as you can see here. And then for those that are non-polar, we have aliphatic side chains that are straight chain or branched carbon chains, and aromatic side chains, which are those ring structures. Now I wanted to add tyrosine over here because tyrosine is also aromatic, but it is polar, and so that's why it shows up on this side over here. Okay, so moving on then, let's talk a little bit about how these amino acids function in the body and how they work in various ways. So one of the ways in which your body makes these non-essential amino acids is through a process called transamination. So I've written here, this is when a non-essential amino acid is not available from the diet. It can be made by the body in a process called transamination. And this is exactly what it sounds. What you're doing is you're taking the amine group off of one amino acid and transferring it to some intermediate that has a carboxylic acid group. In this case, it's alpha ketoglutarate, and forming another non-essential amino acid. So this process generally occurs in the liver. Okay, and this is transamination. Again, you're taking the amine group off one and you're moving it onto another intermediate molecule to create a different non-essential amino acid. And this is really useful for our body because if the body's trying to make a protein that requires glutamate and you don't have glutamate for whatever the reason, your body can make it. And so this is the process of how it does that. Now, this is not done with essential amino acids. Okay, remember our bodies cannot make essential amino acids. This is only done with non-essential amino acids, as I've shown you here. Now, when we want to use amino acids for various different things, there's another process that our body undergoes, and this is called deamination. So in this case, our body is going to be using the amino acid to either make other substances. In this case, we can see here to make ketone bodies, as an example, to oxidize or to create glucose. There are certain amino acids that are ketogenic, and there are certain amino acids that are glucogenic. And so, this is one of the ways in which we can use amino acids to make other molecules. So what this is, deamination, it's exactly what it sounds like. It's the removal of the amino group from the amino acid. And this takes place in the liver, as you can see here, because our liver then has to do something with the ammonia that the NH3 group becomes. And so your liver is your detoxifying organ, and so what it does, once it deaminates the amino group, it goes through the liver, and then it turns into urea and is excreted by the kidneys. Okay, so this is a process that's done in the liver. So if we read this, again, deamination, the removal of the amino group, and the rest of the amino acid that's made up of carbon and hydrogen plus the side chain, as you can see here, is recycled into something else, or it's oxidized for energy. Okay. There are certain amino acids that can be converted to glucose, and only certain others that can be converted into ketone bodies. Okay. And as I said before, your body does not like to necessarily use protein for fuel or for oxidation, but sometimes it has to. And so when it does that, it also undergoes this process where the amino group is removed, and the rest of the amino acid goes into the Krebs cycle. Okay, and as I said, the NH3 group turns into ammonia, which needs to be converted into urea and is excreted by the urine. So we can test how much amino acids our bodies are using by measuring the urea nitrogen excretion. Okay. Okay, so building proteins. We build proteins in our bodies through condensation reactions, and you may have heard about what these are before. We also build carbohydrate molecules with condensation reactions as well. Okay, so essentially what a condensation reaction is it takes the OH group from one amino acid and the H group from the other, and it joins them together. Okay, so the OH group from the carboxy end of one amino acid and the H atom from the amino end of another amino acid joins to form a molecule of water, and a peptide bond is formed between two amino acids, creating a dipeptide bond. Okay, and what we see here for the condensation reaction is these are not amino acids, but I'm just showing you a picture to show you that you end up making water when you join these two together. So again, it's the H group from the amino end and the OH group from the carboxylic acid end to join these two amino acids together. You end up with a dipeptide, or a joint molecule, and water. So hence why this is called a condensation reaction. Okay, and to break down to like a dipeptide bond, so to break proteins into their constituent amino acids, we undergo hydrolysis reactions, and we'll talk about enzymes that do that in one of the next lectures. Okay, so hydrolysis reactions, they break down these dipeptide bonds, and they use water, hence hydrolysis, the breaking up of water. And so the same thing happens in opposite. Essentially, you're adding an OH to one end of the amino acid and an H to the other, so that you end up with two distinct amino acids. Okay, so protein structure then. Now that we've talked about how to make amino acids, we've talked about how to make dipeptide bonds and like how to join two amino acids together. Okay, these are called peptide bonds, and as I said here, these link the amino acids together in condensation reactions. You have two amino acids that join together, they make a dipeptide. And amino acids are joined together from their backbone, not from their side chains necessarily, but I'll show you that there is an exception to some of these rules. Okay, So two amino acids together is a dipeptide. Less than, like a protein chain or an amino acid chain that's less than 10 amino acids is called an oligopeptide. And a polypeptide is what we call a protein that has many amino acids in a row. So in this case, it's 10 or more. And then a protein is one or more polypeptide chains folded into some 3D shape. Okay? Oops, sorry about that. Okay, so let's talk a little bit more about protein structure. So the primary sequence of an amino acid is the primary structure. The primary structure is the initial sequence of amino acids by peptide bonds. So we can see over here, this would be a primary structure over here. So it's the initial protein amino acids that are joined together in one particular strand. Okay? And each of these different amino acids are joined together by peptide bonds. So that's the primary structure. The secondary structure is this alpha helix or beta pleated structure of some proteins. And this is done through hydrogen bonding between the different amino acids. So this creates this secondary structure. The tertiary structure starts to form this 3D structure of a single polypeptide chain. And this is done through hydrophobic and hydrophilic interactions of the particular polypeptide chain and the formation of disulfide bridges between the different side chains. So if we take a look over here, we see this is a hemoglobin molecule and it's comprised of 4 different protein structures. So if you take one of them alone, this represents a tertiary structure of this one strand of protein. Insulin, on the other hand, is made up of 2 strands, okay, so 2 primary strands. And in terms of the tertiary structure, if we look at the blue protein itself, and the fact that between these 2 cysteine amino acids, there's a disulfide bridge. This is part of the tertiary structure of this amino acid. Okay? Then we get to the quaternary structure, which is actually the 3D structure or the interaction of 2 or more polypeptide chains via hydrophobic or hydrophilic interactions or disulfide bridges. Because again, this is the 3D structure of the protein, but it's the interaction of more than one polypeptide chain. So both hemoglobin and insulin, which are polypeptide proteins, they have quaternary structures. Okay, so we can see that over here. In the case of hemoglobin, again, there's 4 proteins. 4 polypeptide chains that are bound together that interact with one another. And in the case of insulin, there's 2 polypeptide chains. And these disulfide bridges between these cysteines in the purple and the blue represent quaternary, like parts of the quaternary structure of the protein. All of the proteins in the body have some type of distinct structure. And based on the amino acid sequence of the primary chain and these other forces that are involved with the secondary structure, as well as the interactions between the tertiary and quaternary structure, they form the protein. And again, this dictates its function. Hemoglobin is a round protein for a particular reason. It allows for the transport of the heme molecule and oxygen and things like that within its structure, so it's meant to be round. There are several factors that can affect the formation of these particular protein structures, which we'll get to talking about for the remainder of the class. So one of those is protein denaturation. This is a change to the 3D structure of the particular protein. So it's not a change to the primary structure of the protein, so you're not actually changing one of the protein or the amino acids within the structure, you're just changing the 3D structure. So something happens to the forces that the side chain, the forces that are dictating the structure of the protein that are brought on by the different side chains of the amino acid. So as we can see here, here is a molecule, a protein that has that has 4 different polypeptide chains, kind of in a 3D configuration. When heat is added, or some type of stimulus of some sort, it unravels the protein. Okay, so you can see here it unravels this protein, and so it changes the 3D structure of the protein. And this is not good. Usually when this happens, it renders the protein non-functional. Okay, and we cannot get back to the way it was before. So why do I have pictures of eggs here? Because the egg white is the protein part of the egg. And you know when you crack an egg open normally, the egg white is like a like a creamy, like translucent type color. It's not pure white. When heat is added to the egg, so you cook the egg whites turn white, and there's nothing you can do to make that egg go back to the way it was before. Even if you remove the heat and cool the egg down, there's nothing you can do. So the proteins within eggs are denatured with heat. And so, this is one example of that. There are several factors that can denature proteins, and this becomes important if we talk about that within the body as well. So heat is one of those factors. Changes in pH, UV, X-rays, agitation, all of these can affect the protein itself, and in some extreme conditions, it can act to denature the particular protein. Just as another example with eggs, when you beat eggs, egg whites a lot, they turn into like a meringue, and there's nothing you can do to get them back to the way they were before. So this is a situation, again, where you're adding some type of agitation to the eggs, and it changes the structure and the function of the particular protein, or the egg white. Why this is important to the body. So as an example, if you have a fever, or if your body becomes too acidic or too basic for whatever the reason, then this can have a profound effect on the function of the proteins in the body. I think the fever is what we can relate to the most, and it's part of the reason why if you have such a high fever, if you spike a high fever, your body does whatever it can to try and bring its temperature down, okay? So you want to take medication to help with that, but your body sweats, then it has chills, and it slows everything down so that it can try and deal with the reason why body temperature has gone up. The same thing when in hypothermia, when your body temperature goes down, your body does what it can to bring up the body temperature. And the reason why our body temperature is so tightly regulated is because at a particular body temperature, this is when our proteins function optimally. If that's changed for too long, then that's not a good thing and our proteins cease to work. Again, same thing with pH, which is why we have a lot of built-in buffers within our body that buffer our bloodstream and things like that, including some of these amino acids that we talk about, okay? And just to reiterate again, the important points about denaturation, this is usually a permanent change within the protein, okay? So it renders the protein non-functional when one of these stimuli are applied. And this is a change to the 3D structure, not the primary structure. So what happens when you do change the primary structure of the protein? Well, this is when we get into this discussion about mutations. So a mutation is different from denaturation because a mutation happens when you alter the base sequence of genes, which alters the primary structure of the polypeptide chain. So we'll actually talk about this one next class a little bit, but recall that in order to make a protein, you start with DNA, and then you have RNA, and then the RNA with the ribosomes, etc., they make the protein. And the RNA is essentially reading a recipe to make whatever protein your body is requiring at the time, right? Now, if there is an issue in the DNA with the protein sequence, then the protein that ends up being made in the end will either not be useful it will not function optimally, it will be adverse, etc. So this is the issue with having some type of mutation. And there are many mutations that we can have in our body, and some of them are in non-coding regions of our DNA, which is totally fine. But if it's a coding region, then it certainly does affect the primary structure of the protein. So what happens when you change an amino acid for a different amino acid, which again is what happens when you have a mutation that could affect the structure of the protein or could affect the making of the protein? You end up with your polypeptide being altered in some way. Okay, so let's say you're substituting one amino acid for another, or you're completely removing an amino acid, like a deletion mutation, for example. Because that particular amino acid is not there, or it's been substituted by another one, depending on what the substitution is or what the omission is, this would affect the secondary, tertiary, and quaternary structures of the protein. So again, let's say a cysteine amino acid was removed or was substituted in. We know cysteine amino acids form disulfide bridges. And so, if there's a cysteine amino acid put in place of another amino acid like glycine or something, which doesn't necessarily have, a vibrant side chain, then the cysteine is going to form bridges with adjacent amino acids, and it's going to create different forces, etc. And so that's going to completely change the structure of the protein itself. And so I've shown you here with sickle cell anemia, here is an example of how the making of your red blood cells could be affected by this particular mutation in the primary structure. And this causes the blood cells themselves to have a sickle shape. Now, people can live with sickle cell anemia because the red blood cells are not non-functional, but they're certainly not functioning optimally, okay? Because remember, the red blood cells, they transport hemoglobin and they're oxygen carrying, etc. The oxygen carrying capacity of sickle cell red blood cells is less. Okay, so those with sickle cell anemia do have issues with oxygen transport, and this is brought on by a mutation to the primary structure of the polypeptide for these red blood cells. Okay, so with that, I will end for today. I'm at 38 minutes, and we will start up again next class with some more discussions about protein.